Hello, everybody. Welcome back to a brand new episode of the Little Dudes Insect Academy podcast. Wait, before we get started, I just wanted to say thank you for tuning into this episode of the Little Dudes Insect Academy podcast and just ask you to please hit the like button below or click the subscribe button if you're watching on YouTube or follow this podcast if you are listening. That really helps the show out. But also, if you want to help us further, then join our Patreon. We have an awesome community that is growing there. Or if you want to make a one-time donation, we are a nonprofit foundation, so head to our website. Both the Patreon and the website will be linked down below. But without further ado, back to the episode. I am super stoked to be here today because we are talking about mites, and I'm here with Sofia. Sofia is a PhD student at Arizona State University, and she studies soil mites. And I'm super stoked to talk about mites because I don't think we've ever had a mite specialist on the show before. So we're going to talk about mites. I'm going to try and keep the mite puns to a minimum. There's lots of opportunity there, but we'll see. So welcome to the show, Sofia. Go ahead and introduce yourself to the people. I'm super excited. Thank you so much for taking the time, but let's go ahead and get started. Go ahead and introduce yourself for us.
Hello, hello. My name is Sofia, and I'm really happy. Thank you for inviting me. And yes, I'm gonna introduce you to the wonderful world of mites. I'm a PhD student. I'm in my last year actually at ASU. So my advisor is Dr. Jay Taylor. He's a mathematician, but it's also a birder. It's like everything. I'm pretty good established now, and I'm just super happy to talk about mice at all.
Yeah, and that's exactly what I'm excited to talk about with you today. So, I mean, let's just jump right into it. I want you to give us a little bit of a background of what mites are. What is a mite? What makes them different? We talk about insects a lot on the show. Of course, mites are not insects. So I want you to break it down for us. What is a mite and why do they make you so excited?
Yes, of course. Let's just start by saying yes, mites are not insects, and you can easily identify them in two groups, right? Insects will have six legs and usually antennae, whereas arachnids, which mites are part of, they have typically eight legs. So if you just count legs, you will find out what kind of bug it is. And if they have more than eight, that could be other type of animals.
Mm-hmm.
But we're just not gonna focus on that right now,
Yeah.
because there's so many. They're equally wonderful. But yeah, mites are arachnids, so they're part of the group that involves scorpions, daddy long legs, which I really like that name.
Mm-hmm.
What others? And
Spiders.
spiders, of
Mm-hmm.
course. Yeah. Um, However, mites are very unique in their body plan, so their morphology is very different from all the other arachnids because they have two parts. So usually we'll see a thorax, an abdomen, and a head. So mites will have only one part in the natosoma, which is all the buccal or the mouth parts, right? And they will have a sterosoma, which is the one that has the legs and all the other organs inside. So we'll just see two different parts, and there's nothing like that in the arachnida, but mites. And we also know that mites are super diverse. Whenever you take a look around, there should be a mite there. You probably won't see them because they're super tiny, so they also have successfully colonized nearly every known habitat on Earth. So we know
Wow.
that there's um, more than 50,000 species so far recorded, but
Wow.
we think that might be like even the 10% of all of the total numbers of mites that could exist today. So yeah, they're very interesting, and um, and they're very diverse, as I said, not only they're predators like most of the arachnids are, but they can be parasites, they can be fungivores. That means that they will feed on fungi or yes, or moss,
Hmm.
and they also can be a yeah, they can feed on nematodes as well. So they have a lot of different branches in the cycle of the of life, and they can be either predators or prey. So it's a lot of fun stuff with mites.
Yeah, that's really cool. Okay, so they only have two segments on their body, is that right? Okay,
Yeah,
yeah, so they've got
yeah.
two segments, four pairs of legs, so eight legs. So a lot different than insects. And then they're also usually very small, is that right? So can you give us like a size range of the typical mite?
Yeah, they can be from 300 microns, so that's microscopic, right?
Yeah.
To 3 centimeters or like half an inch, right? And the most I'll say the most popular or most famous mites there are in the world are ticks because we all know we have seen them in dogs. So those are also mites. They usually classify it apart because of their study is more like a medical
Hmm.
grade, and they have a lot of risk factors, but they're also mites. So, and then we have one of the largest mites that I particularly love. That is the giant velvet mite. I don't know
Mm.
if you have seen them, but usually they come out in the rain, in like the monsoon season or rainy season. After the rains, you'll see them. They're like tiny little red dots that look very hairy. That's why they look like velvety.
Mm.
And they're actually not hairs. They are called setae, but they are very special because they usually live underneath underground, and then they will come active in the spring. So yeah, and also that's another thing that I find very interesting about mice, that usually with tiny little creatures, you will expect a life of just like a week, right? Or like mosquitoes, that is like,
Yeah.
oh, 60 hours,
Hmm.
right? But with mites and in general with arachnids, we have, for example, spiders that can live for like 15, 20 years, right?
Hmm.
So this is the same condition that mites have. There are some mites that can live like for 2 or 5 years. So that's incredibly long for a tiny creature. Well, for my end. So yeah, there are a lot of different sizes in mites, and we actually have two different species of mites living with us
Mm.
right now. So we all have two species that usually live in our foreheads, our eyelashes, and our skin. So
Really?
yes, they've been with us for the longest time, and they are obligate parasites. That means that they really need us to live, and they will feed on the sebaceous, the sebum of the sebaceous glands that are in our pores.
No way.
And yeah, we have two different species that they share their lives together. We have Demodex brevis, that is a shorter one, and Demodex follicularum. So they're so small that we cannot see them through the eyes, but we can see them in a microscope. And
Interesting.
they co-evolve with us to the point that even though they're parasites, they don't really harm us, right? We
Yeah.
never really, we can live all our lives without noticing them. And they're pretty special because there's been studies that track for migration um, in the recent migration in humans, because they will transfer usually from parents to kids or from small circles.
Mm.
So yeah, most populations will share their own mites. And so that's pretty interesting. So yeah, that's our first pet, we'll say.
Yeah. Oh yeah, that's really cool. Yeah, I had no idea. That's really, really awesome.
Whenever you feel alone, you can just
Yeah, yeah, yeah. Think of the mites.
right.
Yeah. Oh man, that's crazy. Okay, so mites do everything is basically what you're saying. Like
Yes.
they do all of the things. Okay, so um they're they live all over the place, and that's crazy that you think we've only discovered 10% or described 10% of mites that are on the planet. I mean, that's crazy to me. Yeah, so lots of mites out there. Are they the most diverse of the arachnids, or do you
Yes.
know? Yeah.
Yes.
Okay. That's awesome. Okay. And ticks are basically very large, parasitic, specialized mites. Is that right?
Right. Ticks are parasitic. They're also obligate parasitic, so they have different life cycles, and within those life cycles, they will go with different hosts, and they can grow almost 30 times their size, the original size.
Mm,
Obviously, what we will see is mostly the females because those are the ones who feed and get engorged, and then um, they will disclose their eggs, which can be like a 3,000 eggs per time. So
wow.
yes,
Wow.
yeah, that's what we see all in the infestations as well. But I just want to mention that that's only one, right? One type of mite
Yeah.
that we have. And we usually, when we think about mites, we think about dust mites, for example,
Hmm.
that are harmful and they make us allergic. Um, or we think about ticks. So we really don't have a good view on mites, but um, we'll see that they can be very, very good for either us, for the soil, and just in general, there are many, many species that they don't really do anything to do with us as humans,
Hmm.
right?
Yeah. So let's talk about kind of the life of a mite. What is the average? Like, how does it differ from some of the other life history that insects have? What is like the life cycle of a mite like, and walk us through the life.
Well, I'm gonna pull up the typical response that we all biologists do is that
Hmm.
it depends, right?
Yeah.
It really depends on the species, and their life cycles and their life history as well. We have in evolution, we have two different terms for life forms. They can be R strategies, which they are rapid reproduce, so they live very short lives,
Hmm.
and we have mites who do that. And then we have the K strategies. So the K strategies are animals or just life forms
Hmm.
that spend their lives mostly in developing, right? So
Hmm.
they gather resources. They can do like we do that. It's care for their youngs, all that stuff, and they usually have longer life cycles. So mites have both of those strategies. It really depends on which type of environment they are and what is their colonized history, right? If either they're parasites or not, if they're free life, etc. So, yeah, there's so many different life cycles within the mites. Um, let's see, for example, the ones that I study, the Oribariid mites, they are called box mites or beetle. I think beetle mites. So they look a little bit like beetles because they're usually brown and like
Hmm.
very their kitten is very thick. So if maybe if you really pay attention when you're like in the grass one day, you could probably see them. It's like the size of a grain of salt,
Wow.
which has leg and legs walking
So you
around.
could still see it with your eye, but they're still
Yes,
really, really small.
we'll have to do what my professor Lupita will say, that we have to put our acarological glasses to see them,
Hmm.
right? Because we have to be very aware of them in order to see them. And yeah, so they live long lives. They live typically lives of like 1 to 2 years. Whereas the astigmatids that are a group that was developed from them, so it emerged from the Oridarids, is an R strategy. So they're very rapid lives. They are usually parasites of
Mm.
birds, and they will live on the feathers, stuff like that. So it's also an interesting topic in evolution of why being a sexual um, we'll say like a line of evolution suddenly is okay, I'm going to diverge into an asexual
Mm.
part, and I will not only be very successful but also diverge into many species. So that's one of the questions of evolution of how sexual or asexual advantages are for, right?
Mm.
And we have a lot of asexual species. Almost the 20% of species in mites are asexual, so that's like huge for
Yeah.
a group of animals. So yeah, I couldn't tell you like this is the life cycle, but you know,
Yeah.
um, yeah, it really depends on every group of mites, and we have different five groups of mites that evolved independently. So
Okay.
yeah,
Yeah, so soil mites are what you work on, right?
right.
So what tell us a little bit about them. What have you sort of found interesting about them, and what makes a soil mite so special?
Yeah, so I really like growing up just looking at the soil. I will, when I was a kid, I would try to cure ants that were, for me, they were sick, right? Like when I was a
Mm-hmm.
very young kid, obviously I did more harm than good to them, but when I
Yeah.
was there, I was really into it. Um, but I always being attentive to the soil. So then I fell in love with mites. So after that, um, I found out that soil plays a vital role in everyone's life, right? In earth ecosystem. So anything that you can think of is related to the soil, right?
Yeah.
Our food, our clothes, everything, the water, the nutrients that we need. So everything is related to the soil. However, this is called the poor's man tropical rainforest. Why? Because we don't know much about the soil. We
Mm.
don't know about soil life. We don't know about the interaction that happened there. Um, so because that, many species have not been described, and a lot of bacteria that live in the soil and fungi that live in the soil is not. Uh, we're not capable of reproducing in the lab. So it comes with their
Oh
own
yeah.
challenges. But also, it's super hard to know what's going on there because there's so many interactions happening. It's a full network. So I think 10 years from now, it's been increasing the interest of biologists into really knowing what's happening in the soil. Also, we know that soil has been severely damaged for pollution, intensive farming,
Mm-hmm.
deforestation, all of that we are facing right now. So I think it's more important now to understand how soil acts, and because we can measure now and we can monitorize what's going on with it, and how to take care of it. So based on that, there's so many different life forms that live in the soil. So we can find almost all the life forms in the soil coming from bacteria, fungi, uh, uh, um, animals, and plants, right?
Mm-hmm.
However, we can classify them in different ways. One way is by the size of the animal. So we have microfauna, which are all the um, microorganisms that live there, like bacteria, fungi, nematodes, rotiferans,
Mm-hmm.
even all of those things. Um, and then we have mesofauna, which is where mites are classified for. They
Okay.
are about like 2 millimeters in length. So things like centipedes, right? Termites, all that stuff. And then we have macrofauna, which would be the known engineers of soil, right? Like termites, like soil worms, and ants. So those would be the biggest size of the fauna, and more a bigger size would be the the animals that live in the ground, like beetles, right?
Mm-hmm.
Even mammals that will search for food, all of that. So based on that, mites are one of the biggest colonizers and the most abundant of the mesofauna. So we can have densities of 10,000 mites in just 1 square meter of soil,
Wow.
especially in forest soils and in tropical soils. So it's pretty enormous of their existence in the soil.
Wow.
And they're also very ecologically diverse, as we discussed. So also soil mites can be have different life forms. So it can be predators, they can feed on animals, they can be parasites, they can even hatch on animals to disperse. So that's another very interesting thing that they do. So because of that, I thought like, okay, maybe I want to do something that not a lot of people has taken into action, which is the study of mites. And specifically, I want to help the soil, right? Because
Mm-hmm.
I consider the soil to be a very important part
Oh
of
yeah.
myself.
Oh yeah. Everybody. I
And
mean, yeah, I
right,
mean,
yeah.
all our food is obviously grown in the soil, and this really came to my attention when I took a biology class, and one of my instructors was talking about the decline of the quality of our soil, even within the last 20 years. And it's depleted by like half in the last 20 years. So I think there's like, um, do not quote me on this, a couple, like 3 meters of soil, across the entire earth. And it was cut down by half within the last like 20, 25 years. So yeah, really alarming, at a very alarming rate. And then obviously soil takes a really long time to be, made, right? It takes a really long time to make new quality soil. So that
Right.
really started, kind of woke me up and was like, yeah, if we don't start doing something, we're going to be in big trouble because we
And
rely on this to grow our food.
right, and every more now than ever,
Mm-hmm.
because we have all this globalization, right? So if quinoa is the one that is gonna be the top of the sales, a lot of crop fields would be full of quinoa. So we only have monocrops, right?
Yeah.
And that really damages the soil population of animals because there's
Mm-hmm.
only one plant that you can feed on.
Mm-hmm.
So yes, that's a lot of alarming, at least for the current time. And I don't think we have learned how to be more aware of that. I don't think there's enough people who are farmers who understand the importance of
Yeah.
changing cultives, and or having um, multiple trees of different species, all that
Yeah.
stuff that we see at school, right? But it's not really translated to what's actually happening with the multinational
Oh yeah.
um, companies and all that. So yes, soy is pretty in risk right now. But if we don't know what's going on first, we cannot tell what changes has been.
Mm-hmm.
So that's also important. And people usually ask me like, "Oh, what's important about this?" Like, nobody cares about mites, right? And it's not dangerous for humans. So what's going on, right? And my answer is always the intrinsic value of all life forms, because
Yeah.
not because we're bigger or we're more advanced in quotes, right?
Hmm.
That means that we're more important than other life forms. So for me, that's what it's just enough. It's sufficient to say, okay, yeah, we want to know all the other forms.
Absolutely.
But in terms of like, okay, I want to get grants, right? I want to get money.
Yeah. Yeah.
um,
Hmm.
so I really have to say, why do we care about mites,
Oh
right?
yeah.
So um, first of all, they being one of the oldest of all terrestrial animals. So the fossils are known from early the Devonic, so around 400 million of years ago. So that's so old.
Wow. Yeah.
um, we were not even, yeah, we're not. Uh, Dinosaurs were not even in the picture if
Hmm.
you think about it. So it's just impossible for us to understand the time that they have lived here, and they haven't changed much. So like
Hmm.
bacteria, they don't need much to live. So I think that's like one of the most amazing parts of it, that they don't really need a bunch of things, um, uh, or a big brain, right, to
Yeah.
develop and to be living to this day. Uh, but also, they take part on the nutrient cycle of the soil, so they're very important for the soil formation. Um, they do is that they usually chew on the leaves, right? So they will break apart all the different plant tissue that would be later available for smaller animals or for bacteria and fungi to colonize. So they will speed up the soil formation and
Hmm.
nutrient cycling. So that's why they're very important. And because there's so many, like um, they're a great source of food for all the other animals, like the ants, the termites that we do care about, right? So yeah, that would be the importance of the mites in the soil. Um, they also do transportation of fungi. So because they have their skin is kind of like sticky sometimes,
Okay.
they will get fungal spores. Even there's some mites that will have pockets for fungi.
Oh really?
That's really unknown. Like nobody knows what they are for at this, um, but they will do that, and they will transport it. So
Hmm.
that's also for dispersion of the microbiota in the soil. So it's pretty interesting how they are, and obviously, as all, I love my parasites. I think we should care about them and we should protect them as well,
Hmm.
because it's thought that every single animal has like at least five parasites, right? And each of those have like a parasite itself. So
Yeah.
we're kind
Yeah.
of like jumping around into a multitude of different animals that we don't even care, we don't even know about. So if we lose one species of insect or one species of mite, we're probably losing more that just one. So yeah,
That's really cool, and that's really interesting to think about. I never really thought of it that way because you mentioned earlier that that's how a lot of mites disperse is they hitch a ride on something that's larger. But then
yeah,
also there's things on the mite as well that the mite may or may not know about, right?
yeah. If
Um,
you're very interested, if you want to be amazed, you can search for a image of a mite hike, hatch hiking another mite. So there are mites that live on mites, and they don't feed on them, but they feed on the bacteria that is in their cuticles, right? So they
really?
will feed on whatever is there, and they just just chilling, right, in their bosses. So it's pretty fun to do that.
Yeah, that's so cool. Um, oh man, that's crazy. Okay, so soil mites, obviously really important ecologically, but tell us a little bit about what you are studying specifically. Like, you're almost done with your PhD. What are some of the cool things that you've been learning and studying with that? And what are the some of the things you've found so far?
Oh, that's very interesting. I'm working right now, but I've been working for 6 years in this project, which is a very exciting project. I'm very happy
Mm-hmm.
and very proud of it. We still don't have much data, so I cannot tell you much.
Yeah.
Um, but it's gonna be great, I think. So I'm focusing on the soil Oribarids, which is a type of mite, right, that
Mm-hmm.
lives in the soil, but in the Madriansky Islands. And Madriansky Islands are mountains that are surrounded by deserts, so um, they're being known for being an islands because they share a lot of traits like the islands that we
Right.
usually
Yeah.
remember in the middle of the ocean, right?
Mm-hmm.
Because they're isolated from each other, but they're still some close apart. So
Mm-hmm.
for example, birds can transport very easily, right? However, there are many species that cannot cross entirely
Mm-hmm.
terrains of just a way different habitat, right? So, thinking about this and thinking about mites, my question was how they differ from one mountain to another mountain, and because the mountain itself also has a range of different altitudes, so we have transitional levels from desert scrub to pine oak, right, to pine. So we have a lot of different environments within the same mountain. So my question at first was, okay, what is the population um, structure of the whole mountain, right?
Mm.
If it's we're gonna find the same species across the mountain, and or if we're gonna find the same species in the same habitats but in different mountains. What's gonna
Mm-hmm.
be the most accurate, and also because we have this complex of islands was it's apart, but it was at one point covered with the interglacial events that happened around 15,000 years ago.
Mm-hmm.
So that's pretty recent. So maybe there was a continuously of pine trees or oak trees that form this kind of a corridor with all the mountains. So I want to see how the population structure is in terms of genetics, right? So how genetically different are the populations in this mountain, and that.
Mm.
So we have more than 50 mountain ranges from South Arizona, right, in Tucson, to North Mexico. So I also want to see that range of how they are in terms of um, elevation, in terms of orientation to the east or the west. We also
Yeah.
have two different deserts there. So there's Sonoran Desert, and we have the Chihuahuan Desert. So they're part of the same region. So I want to see all of that. So I have a lot of questions. Um, and because of how big it is, the project, I've been sampling every year during the monsoons. Um, I chose Oribaris specifically, not because I don't like the other mites or because I don't like soil
Mm-hmm.
animals in general, but
Mm-hmm.
it's because I have to graduate someday, and
You have to
it
pick
would be
one.
impossible, right?
Mm-hmm. Mm-hmm.
Impossible because we have hundreds of thousands of individuals, right, that we found
Yeah.
there. Um, so my main objective is to know the taxonomic uh, surveys. So what mites are there, what species are there. Nobody has looked into even the species that are here. And we have so far 7 species of oribarids listed for the state of Arizona, which is incredibly wrong, right?
Mm,
Um, so also I wanted to do a collection of genetic information, right? So
yeah.
I'm barcoding all the different species I'm finding, and to do a description of habitat association in individual species. I'm also looking at the community ecology and phylogeography of the Madrian or varied assemblages. So,
Mm,
as I said, how they interact with each other, how far away they are in terms of genetics, and um, that's pretty much it. So that would be the start for knowing a little bit more what about the mites that are in the Sonoran Desert. Of course, we have still to look for on the rocks, right? Because we only look for oribarids, and we only look for oribarids that were associated to oak forest. So
mm,
now we have to go deeper, to go to the desert, right? To the basin areas and to go to the washes, all of that. So that's gonna be probably a different project for another person.
yeah.
So yeah, it's a very interesting, and there's so much that you can do with mites in especially in places that haven't been studied yet. So that's pretty much my research in a nutshell.
Wow. Yeah, lots of aspects to your project, it sounds like. So I'm kind of interested how, since they're so small, like what's
Hmm.
the, this seems pretty challenging to me. Are you able to like identify them in the field, or do you just basically like pick up buckets of dirt, bring them back, and hope that you have what you need? Like, what's the process like for collecting that data?
Yes, well, we actually the easiest part is to go in the field and hike. We usually get our binoculars and do some birding while we're there.
Nice.
I've seen a lot of different mammals, and I actually saw a couple of bears the other day, and I was so hyped because I never saw bears in my life. So I was like, oh my God, yes, come. But they were far away, so no worries. um, but yes, we get to camp in those beautiful mountains,
Nice.
and then we collect soil. So we collect soil with a shovel, very high technology, a shovel and a
Yeah.
container. So the first question was how much soil we're gonna take, how much
Yeah.
of each. So we're taking a volume of 5 milliliters, so just half of liter of soil and half liter of liter. So that's what we're using for. And then we take it to the lab. In the lab is where everything starts, is really the hard part because we have to extract the mites and
Hmm.
everything else that lives in the soil. So what we do is if we use a funnel, also very high technology, just like a car funnel, plastic.
Really?
There usually are funnels that are specific for microarthropod caption, but they're too expensive for the same thing. You can just do it in your house. So
Hmm.
that's pretty simple. And you put a light source above. So what's gonna happen is that the light will make a gradient of temperature. So the live bugs that are around there in the sample will migrate to the south because they're looking for moisture, they're looking for air and water, so
Yeah.
they will migrate to the south, and then they will fall into a bottle with ethanol. So once they are in the bottle of ethanol, they cannot climb up, they cannot do anything, so they will pretty much die. So I have killed a bunch of
Hmm.
microarthropods.
Hmm.
You can actually. capture them alive. So instead of putting alcohol, you put just a cloth with water and just keep looking. So even if you're in your house, you can do this. Um, and that's for observing live mites, right? And other animals there are. We usually get a lot of termites, a lot of millipedes,
Mm-hmm.
roly-polies, ants, all that you can find. So after we have that sample, we have to sort them out into groups. So we usually do insects, spiders, like just the major groups. And then
Yeah.
after that, I have to go one by one and look at the each mite and see if
Wow.
I can do it by eye, or I'll have to make a preparation. So I will make a slide. This light goes into the microscope, and I will look at the different characters there with the help of a key. I will do the taxonomic identification, and then we do the extraction of DNA to kind of match if we thought um, it was. It's correlated to the genetic data that we have. The
Mm-hmm.
only problem is that we don't have much of genetic information with mites. If you search in data bank, there's not many. Obviously, there's a lot on ticks, and there's a lot of different just typical research mite species. However, we don't have many for any others, and we haven't found really good matches for most of our animals. So it might be um, the first time that we'll upload that. So yeah, that would be another project to do. So see if they are maybe cryptic species that look the same, but they're actually
Yeah.
different species, right? There's a lot of saying that a lot of mite species are worldwide distributed, but maybe they just don't look different morphologically, but
Yeah.
they're very different genetically. So we're gonna see
Mm.
that as well.
Yeah. So that was actually one of my other questions. How do you sequence, how do you extract the DNA of mites? Because they're so small. Like, I'm kind of interested. How are you able to extract the DNA of like one mite and
Mm-hmm.
not get, it would make sense if you took like a couple hundred mites, right? I feel like that mass would be enough, but then it's
Mm,
mixed together. Like that's not what you're looking for. So
right.
what's the process with that? Like, or have you done any of that yet?
Well, yes, we have tried actually different kits. They are kits that they are sold for DNA and tissue, um, specifically for animals. So we had to find our primers. We had to find
Mm-hmm.
the ones that are working for us. So that's usually the easiest part. However, we found that we had very little amount of DNA
Mm-hmm.
on each side. So we really thought about doing multiple individuals per extraction, but there's that problem, right? Like we're
Yeah.
not sure if those two are the same species. So let's
Right.
just work with one individual. So we work one individual, and there's two ways of doing that. One is the destructive way, so you really macerate it with like a tiny insect pin. So that's all you need
Mm-hmm.
to break out the mite, and then you will run the kit. The other one is non-destructive, so you use enzymes that will destroy uh, the um, the proteins, right? But
Yeah.
without really destroying the outer shell. So that's something we've been trying on. Um, it's a little bit more expensive, but it's also great because you can really use the same individual for doing the identification
Yeah.
taxonomically and genetically. So
Mm-hmm.
you're pretty making sure that that's the same individual, that's the same thing that you're
Mm.
seeing and that you're sequencing. So that's really good, and hopefully, well, more like advantageously, nowadays there's so many different techniques you can use for DNA extraction and DNA sequencing that are very good right now. So I think I got to be in a point in science where we can do this relatively easy and relatively cheap, right?
Mm-hmm.
Um, so um, yeah, and then we had to uh, multiply that because we had so much little amount of DNA. So
Yeah.
what we do is to amplify that DNA using PCR, and after PCR, we send it to the other lab where they do the sequenciation. So yeah, it's usually just one single individual. Sometimes it doesn't work, and
Mm-hmm.
it's crazy because there's some species that we're constantly trying on different things, and we have nothing. And some
Mm-hmm.
do have a big amount of DNA, so it's easy for us. And usually you will use a nanodrop, which to measure how much quantity of DNA is. You cannot do that with mites because there's gonna be so little that you don't even
Right.
um yeah, we would try it and be like, okay, that's not enough.
Mm.
So we thought it was done, but we tried to do it without measuring it, and it actually works. So it's trial and error. Yes, definitely.
Yeah, some of the technology now just blows my mind. It's really exciting. So, moving on to a little more about you. You're at Arizona State right now. But what was your past schooling like? How did you end up deciding on mites? What was some of your what were your other schools like?
Yeah. So I was born in Mexico. I was there my whole life, and I
Mm-hmm.
studied biology. When I was a kid, I wanted to be a veterinarian really bad because I never thought that there were biologists who would do
Mm.
biology. So
Mm-hmm.
I thought like, okay, what is the closest thing um, for animals and to take care of plants and animals? Okay,
Yeah.
veterinarian. So um, um, when I was growing up, I realized that there was no veterinary school in my hometown, so I would have to travel, and yeah, it was like a personal thing where my little sister just was born, so she was 1 year old, so I felt like I wanted to be with her, with my
Mm-hmm.
mom, so I decided to study biology instead. So I was like,
Mm-hmm.
okay, let's just try that, and then maybe I can just jump into veterinary school. But once there, I was like, no, this is what I was meant to be,
Yeah.
because then I think in biology, you see all these different life forms, you see bacteria, you see fungi, you see plants. I didn't know anything about protists, right? So the
Yeah.
first time I was introduced to algae, I was like, oh my God, this is so amazing.
Mm-hmm.
So I had a hard time deciding where to go because I like everything.
Mm-hmm.
We had, well, I did my bachelor's in Sonora, so it's pretty close to here,
Yeah.
to Arizona, right? And we were in all the different clubs that I was like they were existed. I was in that club, the
Oh, that's
the
awesome.
ornithology club, the paleontology, the
Mm-hmm.
uh, all of the ones, and so it was pretty hard for me to decide what I wanted to do. So I did work with different things. I worked with monitoring fauna. Which I really like. So it would be going to put camera traps in the forest and then go back like a week or a month after and just check all the different animals that pass through and just counting them. So that was a very, very good experience for me. That's also when I found out about the mountains in the desert. Because in school you only hear about the arid regions, so the desert, and just um, the scrub and all that,
Yeah.
and you really don't understand that there's maple trees in Sonora. Like what?
Yeah.
So for me that was very interesting. So I started to just kind of jump into that branch. Um, and after that, I was just reading a book, and we had a series of books that were called Science for Everyone. So it was just like a general books about everything. So
Okay.
it's about there's a brain, there's plants, there's all of it. So I read one that was about mites, which was very weird because I didn't see anything about mites in my school of biology,
Mm.
which
Mm-hmm.
it's a little bit weird.
Yeah.
There was one class where they mentioned mites as the invertebrate class. I was like, yeah, these are mites. Okay, next. Okay, yes,
Mm-hmm.
right.
Mm-hmm. Mm-hmm.
Uh, so after that, I was very curious about it. I made um, a summer pasantee in Mexico City, which I work with acarologists, which are specialists for mites. And then after that, I couldn't get it out of my chest. Like I was just really into it, and I looked for people who were interested in that, and that's where I met my current advisor. So he was also my advisor for my bachelor. Um, so we were working remotely. I will work on my lab and then tell him what I found, and then he will tell me what to do, stuff like
Mm-hmm.
that. So because of that, I was already like interested in following through with a PhD or a master's. So after that, I applied for ASU, which has a very good program for PhDs funding, and I got a scholarship. So because I come from a family which not be able to provide for ASU education, right?
Yeah.
Well, it's U.S. education.
Yeah.
Um, so I've been very lucky, and yeah, now I'm here, and I don't know what's next, honestly, but just just try to figure out what I'm gonna do next. Yeah.
Yeah. That was actually my next question. What um you still have to finish the PhD, but like what would be the dream um research or like what is the dream career for you down the road? Like, do you want to keep working with mites specifically, or maybe shift to something new, or
Hmm.
like if you were to have it your way, what would you in like the perfect scenario, what would you want to do?
Yes, like would be yes and no because I love mites. I want to keep working on them, but
Mm-hmm.
also working with mites, I also find a lot of different animals that are very interesting to me
Mm.
and also not very known. So things like isopods. There's one that are called pseudoscorpions that are
Oh yeah.
scorpion-like, and they're
Mm-hmm.
so tiny and they're so
Those are,
fun
and
looking.
they eat mites, I think, right?
Yes, they're
Yeah.
predators, but they don't have the tail. They don't have the poison, right? So it's just I feel like those are winning my heart right now. So I want to
Uh-huh.
work with more the general soil biology and soil fauna than just mites. So that would be um, my take, and I would like to do some kind of um, either bioindication or something that could be more into the practical side of what we can do with that information, right? Um, there's mites that there's some antibodies that are used for ecotoxicology, so absorbing minerals
Mm.
and like cadmium and other.
Wow.
Minerals. So I think that that would be a very interesting part. But I definitely want to do a postdoc. Um, I'm looking for one right now. If you have anyone that is looking for postdocs,
Mm-hmm.
students, let me know. But yeah, I would really want to dive in into the more practical aspect of the soil systems and just keep learning about these. Yeah, either here or anywhere else in the world. I'm just looking um, to find where I can be a good asset, as well.
Oh, that's really cool. And it seems like there's so much to do, like so much opportunity out there for soil biology and microorganisms. It seems so interesting, and yeah, lots of opportunity out there for you, I think. And that kind of leads into my next question. Are some of the what are some other like trips you want to go on, or like places you want to see, things like
Mm,
that? Maybe going and seeing what mites are in other places. Like what what are some other places you want to go see?
wow. Every time I go to a new place, I wonder like what mites are here.
Yeah,
Like
yeah,
every time. So I feel like because of my love for the desert, it would be the next question. Like what
mm,
um it's a little bit more tricky because on sandy soil they're deeper in the soil, so you
okay,
have to find like good good equipment to do that. But I think anywhere that I'll go, I'll find good mites to work with.
mm-hmm,
So yeah, I'm very excited for that. And I'm also interested in knowing more about Sonora in Mexico because um, and the country is very centralized into Mexico City and all the surrounded areas. So we know a lot about there,
mm-hmm,
but we don't know much about the north part. So
okay,
I would love to keep working on um, just increasing the knowledge of mites in the Sonoran area, and of course anywhere in the world like that. I love traveling. Um, our next event, our next conference is gonna be in Chile, so I'm very happy, excited to do that, and hopefully I will be sharing my findings of my research there. So I'm
nice.
very, very, very excited for that. Yes.
Oh wow, um, yeah, very exciting. I'm definitely gonna need to look out for your paper when it comes out.
Yes.
But yeah, super exciting stuff. But I think that's about it. What is there anything else you wanted to mention or talk about, or anything we missed?
More like an invitation for everyone to really look at the underserved and understudied topics in anywhere because usually when I was growing up, I thought everything was already discovered.
Oh yeah.
And when you get into biology, you know that that's not true at all, right? There's always gaps, there's always obscure spaces
Oh
within
yeah.
a field. So just look for that, and I'm very happy doing what I do. I know it's not gonna give me mm, the highest amount of money that it could be I go to the biocontrol of like plagues and all that. It could
Mm-hmm.
be, but that's not really my interest. So I think like within any field you'll find what brings you joy, and I think that's really worth it. So
Oh yeah.
thank you for inviting me. Thank you for doing this podcast. I really like it. I was listening to other uh guests that you had, and everything had so many interesting things to say. So I really appreciate that.
No, I appreciate that you say that. Yeah, thanks so much for coming on here and chatting with us about mites. Super fun time. And for you listening, I will leave Sophia's social media handles in the description so you guys can follow along with her. And um, Sophia, thank you so much for being on the show. This was super fun. So cool to explore the world of mites. It was really, really fun. I learned a whole lot. Um, I didn't really know much about mites. I feel like I still don't because there's so many out there.
Yeah.
But yeah, if you ever feel lonely, just remember those mites that are with you all the time. I don't know if that's comforting or not, but
Yeah, they actually reproduce in your face in the
oh yeah,
net, so that's
yeah.
more comforting.
I get maybe it is, maybe it is. I don't know, but yeah. Thanks so much, Sophia. This was so fun. Thanks so much for taking your time and chatting with me about mites. Um, we talk about bugs a lot, but I feel like mites can be in the bug club.
Yeah, definitely are.
Yeah, they're little crunchy guys with lots of legs, so I feel like they fit. But yeah, thanks so much again. This was super fun.
Yeah, thank you.